Hello, this is Dr. Panigrai. Welcome to my biology classes for NEET exam. Today I will discuss about humoral immunity. As you know that humoral immunity it is the part of acquired immunity, and such humoral immunity also known as antibody mediated immunity. Antibody mediated immunity, or AMI in short. Here the immune cells produce some proteins, and such protein try to neutralize, try to kill, try to inactivate the pathogens or the antigens. That's why the antibody directly involved for the fighting mechanism against the pathogen. That's why it is known as the antibody mediated immunity. So how it works? Let's see. Suppose that these are the antigens. So red spot. Suppose that these are the antigens. Antigen in short, you may write like this: antigen. If such antigen enter into our body, so what is antigen? Any foreign materials which enter into our body, this is known as the antigen. Any foreign materials, like it may be protein, it may be glycoprotein, it may be carbohydrate, or any other materials like pollens or the blood of other organisms. So any materials, if it enter into our body and which activate our immune system, these are known as the antigen. If some antigen enter into our body, actually, as you all know that such antigen, it is recognized by the T helper cell. It is recognized by T helper cell. The T helper cell, by its receptor, they recognize the antigen. So these are the T helper cell and these are the receptor. These are the receptor. These are the antigen. So antigen is recognized by the T helper cell. After the recognition of antigen by T helper cells, the T helper cells get activated. The T helper cells, it get activated. T helper cells activated. Activation of T helper cell. So the activation of T helper cell which switch on the B cells or which activate the B cells or beta cells or bone marrow derivative cells. So the T helper cell which activate the B cells or beta cells. Suppose that this is the B cells. After the activation of B cells, or the beta cells, such B cells, it convert into plasma cells. Suppose that this is the B cells, but this is the nucleus. This is the B cells or beta cells. After the activation, such B cells, it modified into it modified into plasma cells. Such plasma cells secrete, these are the plasma cells having more number of rough endoplasmic reticulum. Such plasma cells secretes antibody. Secretes antibody. These are the antibody. These antibody binds to the antigen. So these are the antibody. Such antibody bind to the antigen. These are the antigen, and it forms the complex. This is the antigen and this is the antibody. These are the antigen, this is the antibody, which forms antigen and antibody complex. Such antigen antibody complex precipitate in the vessels of the blood vessels of the body or the lymphatic vessels of our body, and which accumulate in the body. And such complex, the formation of such complex is known as the agglutination. And such, by the formation of such complex, these complex engulfed by other phagocytic cells or the macrophages, and they digest such antigen antibody complexes, or by the binding of antibody to the antigens become inactive. So in this process, such antibody can remove the antigen from our body. So I hope you understand better.